My name is Raquel Wetzell, and like Marie, I also did my fellowship in the Coral Health and Marine Probiotics Laboratory at the Smithsonian Marine Station. So as Marie already mentioned, the main goal of our lab is to basically identify and treat stony coral tissue loss disease, which is this unknown, potentially bacterial disease that induces bleaching, mucus production, and rapid tissue loss. And it's been around since 2014, where there have been recurrent outbreaks along the Florida Reef Tract, and it's also since spread to many other areas throughout the Caribbean. It affects a wide range of coral species, and this is something we have to consider when we're talking about potential cures. And the main projects that we focus on involve probiotics as the main potential treatment for this disease, which are microorganisms that confer disease resistance to the coral host. So my fellowship actually focused on one of these probiotics, which is OF7M16, and admittedly, it's my favorite probiotic because of its cool color scheme. But my fellowship proposal, I was to characterize OF7M16 and potentially incorporate it into these mixed strain treatments. But what exactly does that mean? So characterization is basically answering these physiological questions about a bacteria under its ideal parameters, or on the flip side of that, less than ideal parameters, and if it can actually survive that. And then mixed strain treatments is basically incorporating multiple bacteria strains together for a broader probiotic treatment. Pretty much based on the theory that if you have a community of probiotics working together to protect the coral organism, it's probably going to have a higher rate of efficacy than a single strain treatment, at least we hope. So for my experiments that I started in January with the start of my fellowship, the first one I did was survival in filtered seawater, my first characterization experiment, which has to do with transporting probiotics to our field site. And these field sites are several hours away from our lab in Fort Pierce, so they can go as far as Miami or Key West or Fort Lauderdale. So we have to have a way to get the probiotic there. So with these experiments, I pretty much put OF7M16 in sterile seawater with no further nutrient inputs. And then I kept track pretty much weekly of the cell counts for this bacteria for 28 days. And after those 28 days, the probiotic was still detectable, and the experiment pretty much went as I expected it to. The only issue was with actually setting up this experiment. I noticed that my bacteria wasn't growing up as properly as it should have. Usually it takes about 8 hours in our lab to grow up a probiotic, but this one was taking me up to 10 to 12 hours, which isn't normal. So then came the fun part of pretty much just playing around with this bacteria and seeing how I could get it to grow better. So my next characterization experiments were actually focused on the growth media to see what sort of media OF7M16 would actually grow up better in. So the main media that we use in general is seawater broth, which is pretty standard. It's an easy recipe to do and it's pretty cheap to make, but for whatever reason, my probiotic wasn't responding to it now as it had before. So I tried a number of different medias, the most important being marine broth, which is actually very similar to seawater broth, but it's a pre-made recipe. So I'm not adding any of my own contents to it. So the flip side of this, I didn't know what kind of salinity levels were with this marine broth. So I had to make pretty much two versions, one with fresh water and no added salt, and the other one with regular seawater, which you measure at 35 ppt, and is pretty standard for the ocean as well. Ran the experiment and tried to see how my bacteria grew in it, and marine broth ended up being the winner, at least the one made with RO water. And when I actually measured it, it turned out to have a 27.2 ppt, which is far less than the 35 ppt we're usually used to seeing. So then I thought maybe I should do more salinity experiments to see if this is what's happening. So with my salinity experiments, the first one I did was in a well plate, which is several channels filled with a minimal amount of media. And that was pretty much a test of broad spectrum of salinities. So I tested 20 ppt, 25 ppt, 30 ppt, and 35 ppt, and ran it as normally, trying to grow up this bacteria. And at first, my results seemed like they were matching what I had gotten with marine broth. So my ideal growth was charted between 25 ppt and 30 ppt. But then I realized these results were actually skewed because of the low volume of liquid in the cells and made them prone to evaporation. So I retested the experiment with flasks, which have a greater liquid volume, and I tested around 26 ppt with 35 ppt and realized there is really no difference in growth between the two salinities. Also, science doesn't always go the way you want, and for some reason my bacteria was just responding as normally again and growing up in the allotted 8 hours. So with that, I decided to just transition away from characterization experiments to kind of give myself a break. And I went into the mixed strain treatment part of my proposal. And with this, I did antagonism assays. So basically the goal of these tests is to see if OM7M16, if I combine it in a mixed strain treatment, is either going to cooperate with other probiotics or inhibit them. So this is like how Yes Marie did too, where we're looking for this zone of inhibition around a spot once we put it on top of the OF7M16. So I tested it first with the other probiotic strains we have in use right now, which is MCH17, SMS1, and MCH142. OF7M16 did not get along with any of them. So those were easy to put aside and allowed me to just dive deep into our isolate library to see what other probiotics I could find that might work well with this. And I had pretty much two criteria for choosing my bacteria. I either wanted ones that were purple, like OF7M16, because of the likelihood that they're going to produce similar compounds, or I wanted bacteria that was also isolated from the host coral, Orbicella faveolata. And the only strain that actually got along with OF7M16 was SS-OFC518. If you look at the plate that I have up, it's probably hard to tell where my spots are for SS-OFC518. It's pretty much indistinguishable from OF7M16. And I did some tests with it, including the salinity well plate experiment, and it responded the same way to evaporation that OF7M16 did. So if you can't tell them apart, they get along with each other and they have the same behavior, it's probably the same bacteria. And these results were pretty much corroborated with what the chemistry lab was doing at the same time with this bacteria. So couldn't really do anything with it. And as I went through the library, I didn't find any other interesting strains that would help me with mixed strains treatment. So I came to the conclusion that I would have to isolate new probiotics. So the good thing is, at around this time, we had also finished our disease transmission experiment with the University of Miami. And there were 3 coral fragments that were left over that didn't develop stony coral tissue loss disease, even after coming into contact with disease fragments. So we figured these might be disease resistant and it would be worthwhile to see if we can isolate probiotics from these fragments. So I took mucus and tissue samples and plated them on seawater agar and minimal seawater agar plates and brew them up for 1 to 3 days. So they ended up looking like this. Not a high amount of diversity that we're looking for, but you never know. There still could be some cool stuff there. So I sampled colonies from those plates and streaked them out. And then from these streaks, I made glycerol stocks that I was able to save away in the freezer, all while coronavirus was happening in the backdrop of this. So I managed to get all of my isolates put away in the freezer. But now I have all these isolates, a good amount of them that still need to be analyzed now to see if there's any potential probiotics in the mix. And then they have to be combined with OF7M16 to see if they will get along and can be incorporated into a mixed strain treatment. In addition to those experiments that need to be done, we also need to do more replicates of OF7M16 in treating diseased coral just to establish how good of a probiotic it really is. And then, of course, those characterization experiments I set aside, still need to do them. So specifically ones that are focused on probiotic transportation and delivery as related to OF7M16. So lots of work and still time to do them, but that's all I have. And I just want to give special thanks to Doctors Valerie Paul, Blake Ushijima, and Jennifer Sneed for being amazing mentors and helping me out through this process. Of course, Eileen and Chris for setting up this whole program, and the rest of the people in my lab.